Hello, I'm your host, Peyton, and welcome to my BAM project. I read Tough Voyages by George R.R. Martin, which, fun fact, if you didn't know, is the same writer of Game of Thrones. Tough Voyages follows a freighter pilot who is moral, strong, and silent. His name is Tough. He's hired by some people to help them board an ancient and powerful ship. When they arrive, however, it turns out to be an Ecological Engineering Corps ship, EEC for short. This ship contains DNA samples of millions on millions of species that are both extinct and alive, and whoever pilots the ship is apparently given godlike powers. All of the people who hired him betray each other, due to all of them wanting to control the ship, which in the end gets all of them killed, except for Tough. Tough now owns the ship, and he believes it's his duty to use it for good across the galaxy. Through his adventure, he helps many planets, populations, and peoples with problems including overpopulation, famine, monster attacks, and many others. He does this all while defending the EEC ship from villainous people. In the end, the plot culminates with Tough helping an overpopulation issue, and he gives them a fruit that has a mysterious powder on it that sterilizes 99% of the people who eat it. Now that my book summary is out of the way, let's get on into our research portion. Quoting my friend and colleague, Bryn Flores, cloning is an experimental technology that involves utilizing DNA samples to make an exact copy of an organism. While the specifics are too complex to get into now, there have been many advances made recently, including cloning primates, which gives us certainty we could clone a human being if we choose so. According to learngenetics.edu, cloning can happen both naturally and artificially. In fact, cloning is something we see more often than we think with identical twins. Currently, there are two main ways of cloning, and those include artificial embryo twinning and somatic cell nuclear transfer. Artificial embryo twinning uses the same approach as natural twinning, but is carried out in a petri dish instead of inside the mother. A very early embryo is separated into individual cells, which are allowed to divide and develop for a short time in the petri dish. The embryos are then placed in a surrogate mother where they finish developing. Again, since all the embryos came from the same fertilized egg, they are genetically identical. The second method is called somatic cell nuclear transfer, SCNT for short. This uses a different approach than artificial embryo twinning, but it makes the same result, an exact genetic copy. In SCNT, the egg cell's single set of chromosomes is removed. It is replaced by the nucleus from a somatic cell, which already contains two complete sets of chromosomes. If you didn't know, a somatic cell is any cell that is not the sperm or the egg.